Hello, everybody. Thank you for checking out my talk here. My name is Daniel Baldassari. I'm an assistant professor at SUNY Oswego, and I'm going to be presenting some hot off the press results that my undergraduate research team and I have been gathering, looking at how living in an urban environment affects the visual signals of northern cardinals. And much of this work here has been done by a wonderfully talented Oswego undergraduate by the name of Christy Derzwiecki. So she has really spearheaded this research, and I'm just here trying not to mess anything up, presenting it to you all. So the basis for this research is the idea that we know humans can affect wildlife communication. Okay, one of the reasons why it's interesting to study interactions between humans and wildlife is not just because we are messing with phenology due to climate change and encroaching on habitats, but we're also just affecting sensory channels by which animals communicate. So some really cool recent examples in marine environments, all sorts of whale species we know that communicate acoustically are disrupted by shipping lanes. There's some really interesting recent research looking at humpback whales and how they avoid shipping lanes and they call much less frequently after ships pass through. This is a beautiful, elegant, amazing new study by Derryberry and her colleagues looking at white-crowned sparrows out in the Bay Area and how when the COVID shutdown was at its height and traffic was greatly reduced, the white-crowned sparrows adjusted their singing behavior. They sang higher performance songs, which showed that they were probably previously being prohibited from doing so by all of the anthropogenic noise. So lots of evidence, these are just recent studies, that humans can affect the ways that wildlife would otherwise naturally communicate with each other. So against that backdrop, we're interested in looking at this phenomenon in northern cardinals, okay? And these are interesting critters to look at this question in because they're pretty widespread. They're in the process of moving northward and westward throughout the United States. They are a dietary generalist. They eat all sorts of things. They're very common visitors to bird feeders, and they eat insects as well, especially during the breeding season. And they seem to be one of these species that really does well in human-dominated landscapes. We can find them in urban parks. We can find them in people's backyards. They really love being in human-dominated areas. And the question is, how is this impacting them? Are they changing in any way, perhaps, that makes them successful in urban areas? And we want to look particularly related to visual signaling. So how does living in an urban environment affect the visual signals, the plumage and other color signals of northern cardinals? And the reason why we think they may be impacted is because of the phenomenon of carotenoid pigments. So carotenoids are pigments that birds cannot manufacture themselves. They need to ingest them through their diet. And carotenoids are present all throughout the feathers and the bills of birds. And cardinals are like the poster child for this. All of that red plumage and the orangish red in the bills, that's all being driven by carotenoids. And we know from some cool physiological studies, as well as some examinations of some of these weird mutant cardinals that show up every now and then, like this yellow cardinal, that the levels of dietary carotenoids, the carotenoids that cardinals are getting into their body through their food, can affect the plumage color that they eventually produce. And so if urban and rural birds have differential access to carotenoids, if they are eating different things, for example, maybe urban birds are eating more birdseed from bird feeders, that may affect the carotenoids that they get into their feathers and affect their resulting plumage color, which may affect their visual signaling system. So where do we do this work? We're here in upstate New York, and we're comparing two cardinal sites. One is very rural. It's Rice Creek Field Station up in Oswego County, owned by SUNY Oswego, there on the left. And then in the middle of Syracuse, right in the heart of central New York, is an urban park called Berry Park. And if you zoom out a little bit, You can really see the difference between these two sites. So Rice Creek is all sort of secondary growth, reclaimed farmland, very rural, no bird feeders in this area, for example. And Berry Park is smack dab in the middle of the city. So two very, very different environments from an urbanization perspective. So these are the two field sites that we're going to be comparing these cardinals at. And what type of data are we collecting? Essentially, we're looking at a couple of different visual signal, what I call patches. We're looking at the back plumage and the chest plumage of males. And so we take feather samples from birds that we capture and we mount them in the lab and do reflectance spectrometry to look at the reflectance profile of the light that's coming off of these feathers. So those top images there on the left, that's the some feathers from the back of a cardinal, and on the right, some feathers from the chest. We also do some image analysis. So we take some standardized photos in the field with color standards in the background. And in this case, we're looking at the male bill color and we're looking at the female underwing. So if you ever see a female cardinal flying, you'll notice that on the underwing coverts and the flight feathers, she has this really beautiful sort of pinkish orange wash. So we're looking at variation in that color patch as well. And what we're going to do, I'll show you how we do this in a minute, is extract a couple of color variables. And these are going to be objective measurements that describe these different colors. And so we either look at the reflectance curves in the case of the feather samples, or we analyze the photos in the case of the bill and the underwing, and we are able to use mathematical models to look at how does the avian visual system, which we know is different from ours, for example, they're tetrachromatic, so many birds can see deep into the UV range. How does that visual system perceive those colors? And we can look at a couple, we're going to look at three color variables here. Hue, which really describes the color, right? Orange versus red, for example. Chroma, which we also sometimes refer to as saturation, okay, which is sort of how pure or how deep the color is. And then brightness, which is basically how much light is reflected by the color. And we're going to compare these different color patches between the urban and rural areas. First thing that we notice if we do that, if we look at our feather samples, the male chest and back feather samples between the rural and urban environments are quite similar. Okay, so the first thing we're going to look at here are just the reflectance curves here. So on the x-axis of this figure, you have wavelengths going from very short wavelengths up to longer wavelengths, and this is the portion of the spectrum that most birds can see, including some UV reflectance in the short wavelengths. And on the y-axis, just how much light is being reflected. And for example, if we look at the red and the green curves here, these are the chest feather samples of the rural and the urban birds. And you can see those curves are almost exactly identical. And a similar thing is going on for the back feather samples here, the blue and the purple curves. Rural and urban birds, very, very similar. And to give you a little bit of a window on how we compute some of these color variables that we're going to look at in a second, this is what these colors look like if you project them into the avian color space. So this is a tetrahedral shape because birds have four cones that can be stimulated by colors. So at the top here we have ultraviolet, for example, short, medium, long wavelength. All these colors cluster down near the long wavelength stimulation, which makes sense. They're mostly reds and oranges. Those are long wavelength colors. And you can see, if we, for example, look at the red and green, again, which are the chest feather samples of rural and urban birds, they're all sitting right on top of each other. Same with the blue and purple, the back feather samples. And to calculate hue, for example, really what we're looking at is the coordinates. Where are we in this color space? That's how we get a number for hue. Chroma, or saturation, is actually how far away are we from the achromatic center where there's no color. So if we're further out toward one cone, we have higher values for saturation or hue. And brightness is just how much light is being thrown off by these colors. So you can actually get that from the reflectance curves themselves. How much light, a brighter color is reflecting more light. So if we look at those three color variables for our chest and back feather samples for our urban and rural males, we're going to look at three different color variables here. So hue, chroma, and brightness. And this is what these look like. So the sort of bright red colors are chest, and the sort of brick red colors here are back. If we look at hue values, comparing rural and urban males, very, very similar. Same with looking at the back plumage color. If we look at chroma, very, very similar story, very similar. No statistical difference here in the chest colors. There is a little bit of a tantalizing difference here in the chroma values for the back plumage. Very, very, very minor difference, and there's some small sample size issues here that we're looking further into, so we're not making too much of this result yet. And brightness as well. Very, very similar. No difference in these feather patches between rural and urban males. Now the story changes a little bit if we start looking at male bill coloration. So now we're looking at photos that we've analyzed in a similar way. We're coming up with these objective color variables, hue, chroma, and brightness. And what emerges is that the urban males seem to have more reddish bills compared to rural males. So we can see that when we look at hue. Okay, so on the left we have our rural males, on the right we have our urban males, and in this case the urban males have a significantly different hue variable. They are falling out in a different location, in different coordinates of that color space, and where they are in that color space is a more sort of orangish, reddish color compared to rural males. If we look at how they compare in terms of chroma and brightness, there's no difference that we see. But these urban males do seem to have a slightly different bill color, and it's more of a reddish orange color compared to the rural males. If we look at the female underwing patches, there's also something kind of interesting going on here. So the urban females seem to have pinker and more saturated underwings. And so we can see that where we look at hue and chroma, the left figure and the center figure here. So if we look at hue first, the urban females here, which are the right box here, their hue values give them a little bit more of a pink color as opposed to sort of a sort of less pinky color. And regardless of the color, if you look at how saturated that color is, they're more saturated in the urban females. So if we move over here to chroma, you'll see that the urban females have significantly higher values for chroma. So their wings have a little bit of a different color. It's more of a sort of deep pink color, and it's more saturated compared to the rural females. Nothing going on with brightness. Urban and rural female underwing patches are equally bright. So there's some tantalizing sort of preliminary results here, and we're continuing to work on this field site, on this system, to gather some more feather samples to increase our sample sizes and have some more images to look at. But it does seem like there might be something going on here where urbanization might be affecting the diet that these birds have, and thus their plumage signals. And this could potentially affect their social signaling and their signaling in mating interactions, which could be very important to reproductive success. One thing that we want to look at to try and figure out exactly what the source of this variation might be is actually measure carotenoids in these birds. So measure circulating carotenoids in the blood of the birds, as well as the carotenoids that are in their feathers. That'll help us figure out exactly what's going on here. And we're also starting a collaboration with some people to look at stable isotopes in feathers, as well as doing some fecal DNA barcoding to actually directly look at what species might be present in the digestive tracts of these birds. So stay tuned for some more results coming out of this system as we learn more about how urbanization might be affecting what cardinals are eating and thus what they end up looking like. And with that, if we have time, I would be happy to take any questions. And I just want to acknowledge some funding from SUNY Oswego and from Rice Creek Associates that made this fieldwork possible. Thank you.